All right, so we're going to start right now by talking about the structure of DNA. That's how we started this unit, so that's how we're going to start this review. So DNA is a molecule that's made up of smaller molecules, and these smaller molecules we call monomers, or building blocks, that eventually get put together to create the polymer of DNA, and we call those nucleotides. So these are the building blocks of DNA, so to speak. Let's draw what one looks like. First thing, it has a phosphate. Attached to that, it has a sugar, and in DNA, this is called a deoxyribose sugar. So I'm going to put an S for sugar, but then specify underneath it that this is deoxyribose. And what that really means, if you want to kind of break it down, D meaning lacking, and then oxy referring to oxygen. It's a ribose sugar that's just lacking an oxygen atom. And then what's going to be attached to that is a nitrogenous base. It's called that because it has nitrogen atoms as a part of it. So this is our base. And underneath here, I'm going to write out our 4 different options. If this is DNA, our options are thymine, which we usually just abbreviate as T, adenine, guanine, and cytosine. And that right there is a nucleotide, these 3 parts, the phosphate, the sugar, and the base. Different base options to make up that base. So what I've drawn here is one strand of DNA made up of multiple nucleotides. So if we were going to circle what in here was a nucleotide, it would be this. So here is one nucleotide. I'm going to label it that. So we can remind ourselves that these are just our building blocks for DNA. Right? Put a bunch of them together and we're starting to get a DNA molecule. But we learned in class that DNA is double-stranded. So there's going to be another strand of DNA over here that's going to pair up with this strand. And it's going to be a complementary strand, meaning that these bases are going to pair with each other. Before we add the second strand of DNA to make a full and complete DNA molecule, it's important to understand what a certain scientist named Chargaff found out. And he discovered this base pairing rule. So let's write out our base pairing rule. And that is that adenine is always going to pair with thymine, and guanine is always going to pair with cytosine, and vice versa. How do we know this? Well, because what holds them together are hydrogen bonds, and the potential for how many hydrogen bonds differs depending on what type of nitrogenous base it is. Adenine has 2 locations for those hydrogen bonds, so does thymine. So they make a perfect pair. They can chemically, physically bind to each other. Guanine and cytosine each have 3 of those hydrogen bond locations. So that's how we, and I say we, I really mean Chargaff, was one of the ones who helped discover that they had this base pairing rule. So now I've drawn the other strand of DNA. So now we have a full and complete molecule of DNA. Double-stranded here. And you can see I've added in these complementary base pairs. So this adenine paired with the thymine over here, thymine with adenine, cytosine with guanine, guanine with cytosine, cytosine with guanine. And keep in mind, too, as we learned later on in the unit, the order, the arrangement of these nitrogenous bases changes the instructions that they give for how to build a particular protein, how to carry out your traits and make you. That all depends here on the inner part, the nitrogenous bases, and the sequence that they are in. So what's not as important for the information of DNA, but is very important for the structure of DNA, is what we call the backbone. So let's go ahead and label the backbone of DNA. And that's consisted of 2 things. The phosphates, which we have on the very outside here, these circles. I'm going to label them P for phosphate, and I'll write a little key over here for us. So P equals phosphate. Oops. Really, these are phosphate groups. Okay, and then we also have our sugar. So here's our sugar. And keep in mind, we're dealing with DNA, so this is going to be deoxyribose. And it's the same over here. It's just upside down because DNA is what we call anti-parallel. One strand is right side up, the other strand is going in the opposite direction. There, so we have all of our sugars and all of our phosphates. If we were to draw kind of a dotted line around this, a lot of times you can highlight all of the sugars and the phosphates, and that's what we would consider the backbone of DNA. It's just for structural support. Let's make sure we can label some of these other really important parts of DNA. So we already labeled our nucleotide. That includes a phosphate, a sugar, and a base. Make sure that you can label all of these as a base. So if I circle this here, what would I label that as? As a base. If I wanted to be more specific, I would label it a nitrogenous base. That would be better. It's just not going to fit on my screen here nicely. And then the other thing that you have to be able to recognize is these hydrogen bonds. That's really important. That's what holds the two strands together. The hydrogen bonds are what get split apart during DNA replication. So we keep referring back to these hydrogen bonds. So let's make sure that we know how to label them. So if I were to circle this right here in the middle, I'm going to abbreviate here H bond for hydrogen bond. And then the last thing that I would want you to focus on is this idea that these base pairs pair up with each other. So if I were to circle this, these are two nitrogenous bases from either strand of DNA that have paired up together. So we actually just call this a base pair. It's a pair of bases. With our adenine and our guanine, we could circle this and consider that a base pair. This thymine and adenine is a base pair, another base pair. So these are some of the very important keywords when labeling a huge polymer, to use that term, of DNA. This whole thing is a polymer because it's made up of multiple monomers. Remember, our monomer here is the nucleotide. So a bunch of these put together starts to build up DNA.